Let's start with something you see every day. Look around you right now. Everything falls into two categories: living things and non-living things. A plant is living, a rock isn't. So what's the difference? What makes something alive? The answer is cells. Cells are the basic units of life. Every living organism has them, and every cell carries out the functions needed for life. Now, organisms are built in two different ways. Some organisms are unicellular. They're made of just one cell. That single cell does everything needed to stay alive. Other organisms, like us humans, are multicellular. We're made of trillions of cells working together. But whether it's one cell or trillions, the cell remains the fundamental unit of life. The story of cell biology starts with some brilliant scientists. Antonie Van Leeuwenhoek was the first person to actually see a living cell under his microscope. Then in 1831, Robert Brown discovered the nucleus, that dark spot in the center of cells. In 1838, Matthias Schleiden, a German botanist, examined lots of plants and realized they're all made of different kinds of cells. A year later, in 1839, Theodore Schwann did similar work with animal cells. He discovered that thin outer layer we call the plasma membrane. Schleiden and Schwann put their ideas together to create cell theory. But something was missing. How do new cells form? Rudolf Virchow answered this in 1855. He explained that cells divide, and all new cells come from cells that already exist. This completed cell theory as we know it today. Let me emphasize the two principles of cell theory because they're absolutely fundamental to biology. First, all living organisms are composed of cells and products of cells. Everything alive is made of cells. Second, all cells arise from pre-existing cells. Cells don't just appear from nothing, they come from other cells through division. These two principles form the foundation of our understanding of life itself. Here's why cells are so important. The cell is both the structural unit and the functional unit of life. It's the smallest thing that can live on its own. Anything less than a complete cell can't survive independently. Even a single-celled organism can do everything it needs to stay alive. Get food, remove waste, reproduce. That's the power of the cell. Cells come in wildly different sizes. The tiniest cells are Mycoplasmas. They're only 0.3 micrometers long. That's incredibly small. Bacteria are bigger at 3 to 5 micrometers. Human red blood cells are about 7 micrometers across. To give you perspective, you'd need over 1,000 red blood cells lined up to span 1 centimeter. At the other extreme, the largest single cell is an ostrich egg. You can hold it in your hand. Nerve cells are the longest. Some stretch from your spine all the way down to your toes. Cell shapes vary just as much. They can be disc-like, polygonal, columnar, cuboid, thread-like, or completely irregular. And here's something important. The shape of a cell usually relates to what it does. All cells fall into two major categories, and this distinction is crucial. First, we have prokaryotic cells. The key feature of prokaryotes is that they lack a membrane-bound nucleus. Their genetic material just floats freely in the cell. It's naked, not enclosed in a compartment. They also don't have membrane-bound organelles like mitochondria or an ER. Eukaryotic cells are more complex. They have a true nucleus surrounded by a membrane. Their DNA is organized into chromosomes inside that nucleus, and they're packed with membrane-bound organelles each, one a specialized compartment doing a specific job. All protists, plants, animals, and fungi are made of eukaryotic cells. That includes us. Prokaryotic cells are represented by several groups of organisms. The main ones are bacteria, which you've definitely heard of. Then there are blue-green algae, also called cyanobacteria. Mycoplasma are unusual prokaryotes, and PPLO, which stands for pleuropneumonia-like organisms. All of these are prokaryotes. Prokaryotic cells are generally smaller than eukaryotic cells, and they multiply much faster. Bacteria come in 4 basic shapes, and these are worth memorizing. Bacillus bacteria are rod-shaped, coccus bacteria are spherical, like little balls, Vibrio bacteria look like commas, and spirillum bacteria spiral like a corkscrew. The shape often helps us identify different types of bacteria. Let's look inside a prokaryotic cell. Most prokaryotes have a cell wall surrounding everything except Mycoplasma, which lacks this wall. Inside the cell wall is the plasma membrane, and filling the inside of the cell is the cytoplasm, a semi-fluid matrix where all the cell's activities happen. The genetic material in prokaryotes is organized differently than in our cells. There's no well-defined nucleus. The DNA is basically naked, not wrapped in a membrane. There's usually one circular chromosome called the genomic DNA. But many bacteria also have plasmid Cs. These are small circles of DNA separate from the main chromosome. Plasmids give bacteria special abilities, like resistance to antibiotics. That's actually a big problem in medicine today. Bacterial cells have a complex protective envelope made of 3 layers. From the outside in, first is the glycocalyx, then the cell wall, and finally the plasma membrane. These 3 layers are tightly bound together and work as a single protective unit. The glycocalyx varies between bacteria. Sometimes it's loose and called a slime layer. Other times it's thick and tough at all. That's called a capsule. The cell wall is crucial. It determines the cell's shape and prevents the bacterium from bursting or collapsing. It's like armor. Scientists use something called gram staining to classify bacteria. Bacteria that take up the stain are gram-positive. Those that don't are gram-negative. This classification is really important in medicine. Prokaryotes have something unique called mesosomes. These are essentially infoldings of the plasma membrane. The membrane just folds inward into the cell. These infoldings can form different shapes, vesicles, tubules, or lamellae. Mesosomes are characteristic of prokaryotic cells. You won't find them in eukaryotes. Mesosomes help with several important functions. They're involved in cell wall formation when the bacterium divides. They help with DNA replication and make sure each daughter cell gets DNA. They assist in respiration if it's energy production, and they increase the surface area of the plasma membrane, which makes the cell more efficient. In some prokaryotes, like cyanobacteria, there are other extensions called chromatophores that contain pigments for photosynthesis. Some bacteria can move around, and they do this using flagella. A flagellum is a thin, whip-like extension that rotates to propel the bacterium. Not all bacteria have flagella. Some are motile, some aren't. A bacterial flagellum has 3 parts. The filament is the longest part and extends out from the cell. The hook connects the filament to the basal body, and the basal body anchors everything to the cell wall. It spins like a motor. Besides flagella, bacteria have other surface structures. Pili are elongated tubular structures made of a special protein. Fimbriae are smaller, bristle-like fibers that sprout from the cell surface. Here's an important point. Pili and Fimbriae don't help with movement. Instead, they help bacteria attach to things. They can attach to rocks and streams, or more importantly, to the tissues of a host organism. That's how many disease-causing bacteria stick to our cells. Now let's talk about ribosomes in prokaryotes. These are tiny structures, about 15 by 20 nanometers. They're made of 2 pieces, a 50S subunit and a 30S subunit. When these come together, they form a 70S ribosome. The S stands for Svedberg units. That's a measure of how fast they settle when you spin them in a centrifuge. Ribosomes are where proteins are made. Several ribosomes can attach to a single messenger, RNA molecule, forming a chain called a polysome. When prokaryotes need to store nutrients, they use inclusion bodies. These are storage granules that lie free in the cytoplasm vein system. They're not surrounded by membranes. You might find phosphate granules, cyanophytene granules in blue-green algae, or glycogen granules for storing energy. Some photosynthetic bacteria have gas vacuoles that help them float at the right depth in water to catch sunlight. Now we're moving to eukaryotic cells, and this is where things get more complex and interesting. Eukaryotes include 4 major groups: all the protists, all the plants, all the animals, and all the fungi. That's a huge portion of life on Earth, including us humans. Eukaryotic cells have several key features. They have extensive compartmentalization. That means lots of membrane-bound organelles, each doing specific jobs. They have an organized nucleus wrapped in a nuclear envelope. They have complex structures for movement and support, and their genetic material is organized into chromosomes. Now, not all eukaryotic cells are identical. Plant cells have cell walls, plastids for photosynthesis, and a large central vacuole. Animal cells don't have these, but they do have centrioles, which most plant cells lack. The cell membrane's detailed structure wasn't understood until we had electron microscopes in the 1950s. Chemical studies, especially on human red blood cells, showed that membranes are mainly made of lipids and proteins. The major lipids are phospholipids, and they're arranged in a double layer. We call this a bilayer. The way phospholipids arrange themselves is really clever. The polar heads face outward toward water from both outside the cell and inside. The hydrophobic tails point inward, away from water. This protects the tails from the watery environment. The membrane also contains cholesterol. Now, the ratio of protein to lipid varies between different cell types. In human red blood cells, it's about 52% protein and 40% lipids. Proteins can be peripheral, sitting on the surface, or integral, buried partially or totally in the membrane. In 1972, Singer and Nicholson proposed what we call the fluid mosaic model, and it's still our accepted model today. The key insight was that the membrane isn't rigid mucin. It's fluid-like. The lipids have a quasi-fluid nature, which means proteins can actually move around sideways within the membrane. It's like pieces floating in a sea of lipids. This fluidity is crucial for many cell functions. It's important for cell growth. The membrane needs to expand. It's needed for forming junctions between cells. It's essential for secretion, where cells release materials. It enables endocytosis, where cells take things in. And it's necessary for cell division. Without this fluid nature, cells couldn't do any of these things properly. One of the membrane's most important jobs is controlling what goes in and out of the cell. The membrane is selectively permeable at Bepelkopf. It lets some things through, but not others. Passive transport happens without using energy. Molecules move along their concentration gradient from high to low concentration. This includes simple diffusion, where neutral solutes just drift across. Water does this too. Said Iyal.App, UEOK. We call water movement by diffusion osmosis. But not everything can just drift through. Polar molecules can't pass through the nonpolar lipid bilayer on their own. They need help from carrier proteins. This is called facilitated diffusion. It's still passive because it follows the concentration gradient. Active transport is different. It requires energy in the form of ATP. Why? Because molecules are being pumped against their concentration gradient, from low to high concentration. A classic example is the sodium-potassium pump that's essential for nerve function. Plant cells and fungal cells have something animal cells don't: a cell wall. This is a non-living rigid structure that forms an outer covering for the plasma membrane. The cell wall does multiple jobs. It gives the cell its shape. It protects the cell from mechanical damage and infection. It helps cells interact with neighboring cells. And it acts as a barrier, keeping out unwanted large molecules. Cell wall composition varies. Algae have walls made of cellulose, galactans, mannans, and minerals like calcium carbonate. Other plant walls contain cellulose, hemicellulose, pectins, and proteins. The structure has layers. Young plant cells have a primary wall that can still grow. As the cell matures, a secondary wall forms on the inner side, toward the membrane. Between neighboring cells is the middle lamella, made of calcium pectate, which glues cells together, and cells stay connected through tiny channels called plasmodesmata that connect their cytoplasm. Now we're going to explore the endomembrane system. This is a group of organelles that work together in a coordinated way. The system includes the endoplasmic reticulum, or ER, the Golgi complex, lysosomes, and vacuoles. These organelles cooperate to process, package, and transport materials. Now here's an important point. Mitochondria, chloroplasts, and peroxisomes are not part of the endomembrane system. Why not? Because their functions aren't coordinated with the other organelles. They work independently. This is a common exam question, so remember, endomembrane system includes ER, Golgi, lysosomes, and vacuoles. It does not include mitochondria, chloroplasts, or peroxisomes. The endoplasmic reticulum, or ER, is a network of tiny tubular structures scattered throughout the cytoplasm. When you look at it under an electron microscope, it looks like a complex maze. The ER divides the cell's interior into two distinct compartments. The luminal compartment is the space inside the ER. The extraluminal compartment is the cytoplasm outside the ER. There are two types of ER. Rough ER has ribosomes stuck to its surface, making it look bumpy or rough. You find lots of rough ER in cells that actively make and secrete proteins. It's continuous with the outer membrane of the nucleus. Smooth ER doesn't have ribosomes, so it looks smooth. Smooth ER is the major site where lipids are made. In animal cells, it also makes steroid hormones. So rough ER is for proteins, smooth ER is for lipids. The Golgi apparatus was first observed by Camille Golgi in 1898. It's named after him. The Golgi consists of flat, disc-shaped sacs called cisternae. Each one is about 0.5 to 1 micrometer in diameter. These cisternae are stacked parallel to each other, kind of like a stack of pancakes. The number of cisternae in a Golgi complex varies. The Golgi has two distinct faces. The cis face, or forming face, is convex and receives materials from the ER. The trans face, or maturing face, is concave and releases finished products. So materials flow from cis to trans. What does the Golgi do? It packages materials for delivery, either to other parts of the cell or for secretion outside. Proteins made on the rough ER get modified as they move through the Golgi cisternae. The Golgi is also the important site where glycoproteins and glycolipids are formed. That's why the Golgi stays in close association with the ER. They work together. Lysosomes are membrane-bound vesicles that form in the Golgi apparatus. They're packed with digestive enzymes called hydrolytic enzymes. These include lipases that break down fats, proteases for proteins, and carbohydrases for carbohydrates. These enzymes work best at acidic pH mice. The inside of a lysosome is acidic. Together, these enzymes can digest all the major types of biological molecules like carbohydrates, proteins, lipids, and even nucleic acids. That's why lysosomes are sometimes called the cell's digestive system. Vacuoles are membrane-bound spaces in the cytoplasm. They contain water, sap, and excretory products, basically materials the cell doesn't need. The membrane around a vacuole is called the tonoplast. In plant cells, vacuoles are huge vlioles. They can take up 90% of the cell's volume. The tonoplast actively transports ions and materials into the vacuole, even against their concentration gradient. So concentrations inside the vacuole can be much higher than in the cytoplasm. In amoeba, the contractile vacuole handles osmoregulation and excretion. In many protists, food vacuoles form when they engulf food particles.
Way more convenient to productions. I'm going to finite organelles. Unless specifically stained, they're not easily visible under a regular microscope. The number per cell varies mitochondrially. It depends on how active the cell is. More active cells need more mitochondria. They're typically sausage-shaped or cylindrical. Size-wise, they're about 0.2 to 1 micrometer in diameter, with an average of 0.5. Length ranges from 1 to 4.1 micrometers.
Mitochondria have a complex double membrane structure. The outer membrane forms a smooth boundary. The inner membrane folds inward, forming structures called cristae. The inner compartment is filled with a dense matrix. Those cristae are important. They massively increase the surface area for chemical reactions. Each membrane has its own specific enzymes. Now, what do mitochondria do? They're the sites of aerobic respiration, where the cell produces ATP. That's the energy currency of the cell. That's why we call mitochondria the powerhouses of the cell. Here's something really interesting. The matrix contains its own circular DNA molecule, some RNA, and 70 S ribosomes, the prokaryotic type. Mitochondria can even divide by themselves through fission. This suggests they might have evolved from ancient bacteria. Plastids are found in all plant cells and in a group called euglenoids. They're large organelles, easily visible under a microscope. They contain specific pigments that give them different colors. Based on these pigments, we classify plastids into 3 types. Chloroplasts are green because they contain chlorophyll. Chromoplasts have yellow, orange, or red colors. And leucoplasts are colorless. Each type has a specific function. Chloroplasts contain chlorophyll and carotenoid pigments. These are responsible for trapping light energy, which is essential for photosynthesis. Chromoplasts contain fat-soluble pigments like carotene and xanthophylls. These give parts of plants their yellow, orange, or red colors. Think of carrots or autumn leaves. Leucoplasts are the storage plastids. Amyloplasts store carbohydrates as starch. Potatoes are full of these. Elaioplasts store oils and fats, and aleuroplasts store proteins. Most chloroplasts in green plants are found in the mesophyll cells of leaves. That's the tissue between the upper and lower surfaces. Chloroplasts come in various shapes, lens-shaped, oval, spherical, discoid, or even ribbon-like. They're about 5 to 10 micrometers long and 2 to 4 micrometers wide. The number per cell varies a lot. A single-celled alga like Chlamydomonas has just 1 chloroplast, but a mesophyll cell might have 20 to 40. Like mitochondria, chloroplasts are double membrane bound. The inner membrane is relatively less permeable than the outer one. The space limited by the inner membrane is called the stroma. In the stroma, you find organized, flattened sacs called thylakoids. Thylakoids are arranged in stacks that look like piles of coins. These stacks are called grana, and a single stack is a granum. Flat tubules called stroma lamellae connect the thylakoids of different grana. So the thylakoid membranes enclose a space called the lumen. The stroma contains enzymes for making carbohydrates and proteins. It also has circular DNA and 70 S ribosomes, a template again, like mitochondria, suggesting an ancient bacterial origin. The chlorophyll pigments are in the thylakoids. That's where light is captured. Ribosomes were first observed under the electron microscope by George Palade in 1953. They appear as dense, granular particles. Ribosomes are made of RNA and proteins bound together. Here's an important point. Ribosomes are not surrounded by any membrane. They're one of the few organelles without a membrane. There are 2 types of ribosomes. Eukaryotic cells have 80 S ribosomes made up of a 60 S and a 40 S subunit. Prokaryotic cells have 70 S ribosomes with 50 S and 30 S subunits. Now, S stands for Svedberg's unit distribution. When ECO, it's the sedimentation coefficient. This is a measure of how fast particles settle when you spin them really fast in a centrifuge. It's indirectly a measure of their density and size. Both types of ribosomes have 2 subunits that come together during protein synthesis. The cytoskeleton is like the cell's internal scaffolding and muscle system combined. It's an elaborate network of protein filaments throughout the cytoplasm. The cytoskeleton has 3 main components. Microtubules are the largest, microfilaments are the smallest, and intermediate filaments are, intermediate in size. What does the cytoskeleton do? It provides mechanical support, keeping the cell from collapsing. It enables motility cell movement, it maintains the cell's shape, and it helps transport materials around inside the cell. Without the cytoskeleton, cells would be like water balloons with no structure. The cytoskeleton gives cells their shape and helps them move and function properly. Cilia and flagella are hair-like structures that stick out from the cell membrane. Cilia are small and work like oars, either moving the cell itself or moving fluid around the cell. Think of the cilia in your respiratory tract that sweep mucus upward. Flagella are longer and are mainly responsible for moving the cell through its environment. Sperm cells have flagella, for example. Now, bacteria also have flagella, but their structure is completely different from eukaryotic flagella. The internal structure of cilia and flagella is fascinating. They're covered with plasma membrane, and inside is the core called the axoneme. The axoneme has microtubules running parallel to the long axis. The arrangement is very specific. It's called the 9+2 array. There are 9 pairs of microtubules arranged in a circle, plus 2 central microtubules. The central ones are connected by bridges and enclosed in a central sheath. 9 radial spokes connect the central structure to the peripheral doublets, 1 spoke to each doublet. The peripheral doublets are also interconnected by proteins called linkers. The whole structure emerges from basal bodies, which are similar to centrioles. This 9+2 arrangement is remarkably consistent across different organisms. The centrosome is an organelle that contains 2 cylindrical structures called centrioles. These are surrounded by amorphous paracentriolar material. The 2 centrioles lie perpendicular to each other at right angles. Each centriole has what we call a cartwheel organization. It looks like a wheel when viewed from the end. Each centriole is made of 9 evenly spaced peripheral fibrils of tubulin protein. Each of these fibrils is actually a triplet, 3 microtubules fused together. The adjacent triplets are linked to each other. There's a proteinaceous structure in the center called the hub, and it's connected to the peripheral triplets by radial spokes. What do centrioles do? They form the basal bodies of cilia and flagella. And during cell division in animal cells, they help form the spindle fibers that pull chromosomes apart. That means the nucleus as a cell organelle was first described by Robert Brown way back in 1831. Later, a scientist named Fleming studied the material inside the nucleus. He noticed it could be stained with basic dyes, and he gave it the name chromatin. The nucleus is surrounded by a nuclear envelope made of 2 parallel membranes. Between these membranes is a space of 10 to 50 nanometers called the perinuclear space. This envelope forms a barrier separating nuclear contents from the cytoplasm. The outer membrane is continuous with the endoplasmic reticulum and has ribosomes on it. At various points, the 2 membranes fuse to form nuclear pores. These pores are crucial, meaning they're the passages where RNA and protein molecules move in both directions between the nucleus and cytoplasm. Most cells have 1 nucleus, though there are exceptions. Some mature cells even lack a nucleus entirely, like red blood cells in mammals or sieve tube cells in plants. Inside the nucleus, the nucleoplasm or nuclear matrix contains 2 main things, the nucleolus and chromatin. The nucleoli are spherical structures. They're not surrounded by membranes. Their content is continuous with the rest of the nucleoplasm. The nucleolus is where ribosomal RNA is made. It's the ribosome factory. Cells that make lots of proteins need lots of ribosomes, so they have larger and more numerous nucleoli. When a cell isn't dividing, we call this interphase. The nucleus has a loose, indistinct network of chromatin. But during cell division, this chromatin condenses into structured chromosomes that we can see clearly. What's chromatin made of? It contains DNA wrapped around basic proteins called histones, plus some non-histone proteins and RNA. Here's something amazing. A single human cell has approximately 2 meters of DNA. 2 meters! And it's all packed into 46 chromosomes, organized as 23 pairs. Chromosomes are only clearly visible when cells are dividing. Each chromosome has a primary constriction called the centromere. On the sides of the centromere are disc-shaped structures called kinetochores nisca. These are attachment points for spindle fibers during cell division. The centromere is what holds the 2 sister chromatids of a chromosome together. We classify chromosomes into 4 types based on where the centromere is located. Metacentric chromosomes have the centromere right in the middle, forming 2 equal arms. Submetacentric chromosomes have the centromere slightly away from the middle, creating 1 shorter arm and 1 longer arm. Acrocentric chromosomes have the centromere close to one end, so you get 1 extremely short arm and 1 very long arm. Telocentric chromosomes have the centromere right at the tip, its terminal. Sometimes chromosomes have a non-staining secondary constriction at a constant location. This gives the appearance of a small fragment we call a satellite. These are important features for identifying specific chromosomes.
Let's wrap up with the key concepts you need to remember. First and most important, the cell is the fundamental structural and functional unit of life. This is the core principle of biology. Cells come in two major types, prokaryotic and eukaryotic. Prokaryotic cells lack a membrane-bound nucleus and don't have membrane-bound organelles. Eukaryotic cells have a true nucleus surrounded by a membrane and contain many specialized membrane-bound organelles. Remember the major organelles and what they do. Mitochondria are the powerhouses that produce ATP through aerobic respiration. Chloroplasts carry out photosynthesis in plant cells. The ER synthesizes proteins and lipids. The Golgi apparatus packages and modifies materials. Lysosomes contain digestive enzymes. Ribosomes are where proteins are made, and the nucleus is the control center containing the cell's genetic material. Each organelle has a specific job, and they all work together to keep the cell alive and functioning. That's the beauty of cell organization. It's a perfectly coordinated system where every part has its role.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.